And on the right side, you see the most extreme form of therapy, and that would be lung transplantation, which sometimes needs to be done for some. And on the left would be ways to ameliorate CF by attacking the very fundamental problem, such as replacing the gene. Addressing the basic defect has another approach that's similar to replacing the gene, which is to use drugs that turn CFTR back on or bring more CFTR channels to the surface, depending on the nature of the problem. And we'll take a look about those type of therapies and where they stand with much of the rest of the talk. But it would be remiss to say that, again, to remind you that many of the advances that have resulted in improvement in the lives of CF patients and improvement in the life expectancy of CF patients have been treatments to address the abnormal ion transport environment or the mucus secretion problem. And these could be therapies like pulmozyme and hypertonic saline, or to address the infection and inflammation and tissue destruction. And these would be therapies that CF patients use every day, such as azithromycin or large high-dose ibuprofen and inhaled antibiotics such as Toby and Casein and others. So again, much of the rest of the talk is going to be talking about what's being advanced in terms of rescue of the CFTR protein. Well, to understand how we might rescue the CFTR protein, you need to have a firm understanding of the molecular pathogenesis of CF and how we might intervene to correct the abnormal protein. And what's been done is to break up CFTR mutations into major categories and then develop therapies based on those categories. One common form of CF is caused by class 1 mutations, which is caused by premature termination codons, or nonsense mutations, or other mutations such as insertions and deletions that cause major disruptions of the CFTR gene. And that's what's called class 1 mutations, where little or no CFTR protein makes it to the surface. And you can see the X at the bottom of the screen where the nucleus is, where CFTR channels are first being made. Class 2 mutations include the most common mutation, the delta F508 mutation, or F508del. These channels are misfolded in the cell, recognized as being misfolded and degraded in a compartment called the proteasome before they ever reach their active site at the cell surface, before they're able to even think about doing their job. There are other mutations that reach the cell surface but are defective for various reasons. That includes the class 3 mutations, which are like a channel that's locked shut. They're there at the surface, but that engine that we showed on some of the videos that functions to move those CFTR channels open and closed, that engine is completely broken. The channel does not gate, and therefore it doesn't move the chloride and bicarbonate and thus the salt. There are other mutations such as class 4 mutations. This would include the common R117H mutation, but many others. That the channel, the CFTR channel gates okay, but the channel is partially obstructed so that chloride and bicarbonate doesn't flow through the channel as fast as it should. And then there are other more rare mutations, but important ones, including class 5 mutations, where the protein is made wrong because of a splicing defect, or where the CFTR channel doesn't reside at the cell surface very long. The CFTR channel needs a signal to stay on the surface, and when that's broken, you get a class 6 mutation where it simply doesn't live on the surface for very long. And we now know that there's therapeutic approaches being advanced that could cover many of the CFTR mutation groups that we've shown. For instance, nonsense mutations, one form of class 1 mutations, can be addressed with agents that cause translational read-through or cause the cell to ignore an abnormal stop sign. We'll talk about each of these in a bit greater detail. Another group is called the F508del processing correctors, or just correctors for short. This includes the agent VX809 that's been in study and causes the CFTR protein to fold up more correctly and therefore migrate through the cell and reach its active site at the cell surface. And there are agents called CFTR potentiators that function to activate or potentiate the channel and allow it to function more normally. And those are really being advanced mostly for mutations that are at the cell surface but don't function to gate normally. This agent really includes the agent VX770 or ivacaftor, which really accelerates gating. It accelerates that opening and closing motion we've seen, and it has now been shown to be effective in patients with the very common class, or the most common class 3 mutation, G551D. But it's now being studied in these other groups of surface localized CFTR channels. So we're going to take a closer look at the progress that's being made with CFTR potentiators and efforts to address the G551D mutation. So here we're going to see a video of that channel that's locked shut, and then what a drug like a CFTR potentiator, like ivacaftor, would do to that channel. So again, here's the channel, and it's locked shut. You can see the chloride and bicarbonate doesn't move. And then when the drug comes in and binds CFTR, which we now think that it does, you can see the gating starts, and then the chloride bicarbonate starts to move. And again, you can imagine that would allow that fluid to become more normal and the mucus to begin to transport. And we know that when in phase 3 testing, that allowed ivacaftor to be approved for patients with the G551D mutation who are 6 and above, that there was really very large improvements in lung function that was surprisingly rapid. You can see it was observed here at the earliest time point, the 2-week time point in this study, and the magnitude of that benefit compares very favorably with other therapies that are now being used commonly for CF patients. And what we've seen is a very rapid uptake of this drug among patients with the G551D mutation. And like other treatments, we've seen a significant improvement in the frequency of pulmonary exacerbation, certainly an important marker for what we'd expect a drug to do. And it was accompanied with a remarkable improvement in sweat chloride. The sweat chloride improvement, as you can see here, was about 48 milliequivalents. We now know in post-approval studies that many centers are participating in, including Stanford, that improvement in sweat chloride was recapitulated exactly to the T, within a milliequivalent of that estimate. And what this is important because the sweat chloride test is one of our cardinal markers for how well CFTR is working that we can measure in people. And in fact, when you drop a sweat chloride by this much, the average sweat chloride in patients in this study was below the traditional diagnostic range of CF. And while we don't claim that these patients are normal or that their CF is cured, that would certainly be overstating the case. It's certainly important that their CFTR function is down into the subdiagnostic range and conferring those improvements that we've seen in lung function and exacerbations among a number of other markers. The way this is a model of how some scientists are thinking about using this case as a roadmap for what needs to be done next to bring these type of CFTR-directed therapies to many more patients. On the left side, you can see the CF in its most severe form, or what we call pancreatic insufficient CF. CF so bad that you need to take pancreatic enzymes on a daily basis. On the right side is a non-CF, or essentially normal individuals. You can see right next to them are carriers. And if you look at the sweat chloride test, it's not a perfect line, but it's this curved line that you can see where sweat chloride really drops quickly on the left side as even small amounts of CFTR function confer improvements in those phenotypes. And we can use the evidence from the ivacaftor studies in patients with G551D to begin to get a sense for how well we need to do, how much function needs to be obtained in both the laboratory and the clinic to result in an improvement that patients and families would be happy with. For instance, we know from earlier testing of ivacaftor that the various doses that were tested land pretty much on this line when we measure CFTR function with a couple of different ways, either sweat chloride testing or nasal potential difference testing. And we also know that it was these moderate doses and greater that resulted in improvement in lung function. And so that gives us kind of some zones of interest where we can expect improvements to be seen, certainly in early phase testing, which is generally small groups of people, say 10, 20, or 30 individuals, to know if a drug is safe and can be moved forward. And so for this, in this example, and certainly there's debate exactly where these cutoffs go, but just using this as a representative example, you can see that we think we have to get about 30% CFTR function to really make a huge difference in the way patients live. And yet, if we don't accomplish that, but are still able to confer, say, 10% or 15% or 20% improvement, that we should still expect to see some measure of improvement among CF patients. And that's one marker that's being used to help drive science and know how well we need to be. We can envision what it would be like if CFTR function were returned in this way. And again, in this video, we'll see what would happen to the surface if CFTR comes back, moves salt and chloride and bicarbonate, and allows the mucus to break up and move. And so in this quick video, we'll see, again, that surface liquid starting to be reassembled, and the cilia then allowing themselves to beat and then begin to clear out the mucus and have a more normal situation. And that's what we think happened with the administration of ivacaftor to patients with the G551D mutation, and allowed the lung function then to improve in that way. Well, certainly, while that's a successful vignette, it's certainly not enough. That's only 4 to 5% of CF patients worldwide, and now there's considerable effort to advance these type of strategies to more CF patients. So while it's a remarkable advance, it's also a reminder of the challenge ahead. And the initial steps to try and expand the number of patients that could benefit from a drug like this are relatively straightforward. Here you can see a grid on the bottom of the slide just reminding us that 4% is not enough. But what's being done now is to first take the patients that are below 6 and study the same drug in the same mutation groups and look to see if it's advanced there. And that would add, we think, about 0.5% of more of the population. There's also a very closely related to the G551D mutation, a group of very rare mutations that make up about 1% of the CF population, where we think ivacaftor works very, very similarly and equally as well. And so those patients are now being enrolled in a study to look at the group of gating mutations, not just the G551D mutation. And many centers are enrolling in that trial. Well, what about other mutations? So if you look at this slide, we've talked about the one that's on the surface and far left that's noted by class 3. But there's good evidence now in the laboratory that CFTR potentiators, by stepping on the accelerator of gating, could overcome problems that aren't necessarily related to gating. Because the CFTR channel, when it gates, it's not gating at full speed. So if you could take that CFTR channel and take it to full speed, maybe you could make up for the fact that it doesn't move chloride and bicarbonate as efficiently otherwise. And that's the strategy that's now being tested in patients with class 4 mutations, or the R117H mutation. So what does that look like? Returning to these videos that hopefully reinforce what I'm saying and let you visualize it. Here's a channel that's moving some chloride. You see some of the chlorides moving through, but essentially gating can be put on overdrive and make up for the fact that the chloride movement is relatively slow. And what you can see, hopefully, is that the gating is about twice normal speed because the accelerator that's posed by a CFTR potentiator is being employed. So this is now being studied in yet another study that's specifically enrolling patients with the R117H mutation. And that was chosen because it's the most common class 4 mutation, and that's now enrolling in many sites across the country, but not just in the U.S., but in Europe and Australia, to answer the question whether or not these other groups of mutations can also benefit. And if successful, we begin to start to see that we're making a little bit bigger dent in the number of patients that could be eligible for a CFTR potentiator. Well, another approach is to try and abandon this mutation by mutation by mutation strategy and group people based on how they're doing instead of what mutation they have. At some point, certain mutations are so rare we can't imagine doing large studies to really understand how a drug might work in these rare mutations. Your families might be among those groups. So another way to do this is to group patients by how well they're doing and hopefully attack larger groups of people. One way to do that is to take the patients that generally have a milder form of CF, because if you have a more mild form of CF, you can infer that many of those patients will have a CFTR channel that's present at the surface and working, but just not working as well as normal. And so again, by stepping on the accelerator of gating, you could make up for whatever abnormality that is. And so that's a strategy that's being looked at in a study that's called an N of 1 study that takes a patient, makes sure that they're in that group of individuals that has partial CFTR function, and then applying ivacaftor. And that's a much smaller study that's being done at the University of Colorado in Denver. But again, the basis of this is that many of the mutations on the right side of the screen have partial activity, shown by the bar graph. You can see the mutations are listed on the bottom, and the CFTR function is listed on the y-axis, and the ones that are on the right are the mutations that have partial activity to begin with. Those patients generally have a more mild form of the CF, although that rule isn't absolute. And we know that the more function that mutation had to begin with, then the more that stepping on the accelerator of gating benefits that mutation. And so that's the strategy that's being used to enroll these individuals. So what about mutations that aren't at the surface that have other problems? Well, this is actually how I got my start in the CF business, a research business that is, and we're thinking about treatments that cause the translational read-through or premature stop codons to be ignored to allow a protein to reach the cell surface. That's been a strategy that's been employed, for instance, by PTC Therapeutics for the agent Adalurin, or formerly known as PTC124. I'll show a video of what this strategy looks like. This time, we're zooming in on the protein machinery that we saw in one of the first videos I showed, where we see the ribosome functioning to translate the message that the genes make. You see that strand that's running from the top left to the bottom right of the screen is the message that encodes our genetic code. And the machine of the ribosome that we saw before is there like a bead on the strand. And it's going to function to make the CFTR protein. Here, again, it's moving along the strand, and you can see that CFTR protein's being built. But in this case, it runs into a stop sign that shouldn't be there, a premature stop codon, and essentially the ribosome breaks off, stops doing its job, and you're left with this partially active truncated form. And in other research, we know that the mRNA message, that message just kind of disintegrates and is destroyed by the cell, since it doesn't encode a full-length protein. So those are two problems that need to be addressed by this strategy of translational read-through to try and get those stop codons to be ignored. Well, we now know from work by PTC, but also work in the laboratory by a number of groups, including our own, that this process can be ignored. And what this would look like here, again, is the ribosome, we'll see, reaches the stop codon, and instead of breaking off, just slows down until a drug can come, bind the ribosome, and cause that stop sign to be ignored, and then translation to continue to ensue and make full-length proteins. So we'll see that in this video here. Again, you can see the translation process happening. A drug hits at the place of the stop codon that allows that process to be ignored, and now a full-length protein is made to do its job. And in addition, we think that process could even stabilize those transcripts to allow more and more translation to occur, stabilize that message to allow more and more protein to be made and benefit to be conferred. So because premature stop codons are about 10% of CFTR mutations, that's another approach that could add a group of 10% of individuals. And this is work in our lab that shows this is indeed possible. We're using agents that are derivatives of a common antibiotic, gentamicin, that is known to cause read-through at very, very low levels. And we've been able to optimize those drugs to improve the level of read-through. And we're getting to a point where it's close to being enough that we could expect it could make a difference for CF patients. And in addition, we know that we can perhaps, even if it's not enough, but it's some, we could use a potentiator to step on the accelerator of those channels as well and further accelerate that. And so that's research that's in the laboratory at present, but attempting to be advanced to individuals. I'm going to spend the rest of my time talking about approaches for the most common CFTR mutation and return to the F508 deletion mutation and look at correctors that try to improve the folding of CFTR. And this includes the agent, the VX809. Well, the correction of F508 is a very important target, but it's also a challenging one because it exhibits multiple problems. It has impaired cellular processing, which means it's recognized as misfolded and removed before it ever reaches its active site at the plasma membrane. Even when some escapes that process, we have a quality control process that removes those proteins, but sometimes a little bit of F508 protein escapes and makes it to the surface. But the material that does make it through there doesn't gate normally either. So it's almost a mix of two problems. And then yet another problem that it has is that when it makes it to the surface, if it's misfolded, the cell also patrols proteins that are misfolded that are at the cell surface and will remove misfolded protein from the surface. So we call that half-life when it doesn't live, that it has a shortened half-life when it doesn't live at the cell surface for very long. So now we're going to see a video, returning back to one of the first videos that I showed, of what it might look like in the cell when this processing defect is present. And what we'll see here is a CFTR is made, it's inserted into the membrane, but it looks different than the CFTRs you've seen before in that it's bigger and misfolded and globular. And therefore, it's going to be removed from the membrane before it ever migrates to the surface. Even when it does migrate to the surface, it's not going to have the function that we expect it to have, like a normal channel would. So here's that ribosome again, and it's inserting the CFTR into the membrane. And when it completes its job, you can see that is misfolded, and they're in color encoded here as black because they're bigger than normal and more misfolded, and they don't go very efficiently into these vesicles. And occasionally a channel will escape and make it to the surface, but the density of those channels, the number of those channels at their surface is very low. And you can see that bottom of that channel is misfolded because the area that controls its gating is not working well, and therefore it doesn't move very much chloride or bicarbonate. And that's what we envision patients with the F508 mutation suffer because of that deletion in their protein. Well, what would this look like in the era of a two-drug treatment that would use drugs to correct this process? One would be a corrector to cause it to fold up more normally and replace the missing phenylalanine, the interactions that phenylalanine normally does to allow that protein to fold up more normally. and then allow essentially at least some channels to reach the surface. And then you're going to see a second drug, such as resembled by the potentiator, ivacaftor or VX770, to then step on the gas on those now appropriately folded channels, but are relatively few in number because none of these drugs are even close to perfect. So let's take a look at that process. So here's a channel, but at the time of the folding, that drug binds and then allows that CFTR channel to become a more tight structure, a more folded structure. And now you can see these yellow channels achieving to the membrane. And therefore, even though they're not quite as the number that we saw in the normal situation, they do migrate in greater number to the surface, insert into the plasma membrane, and serve there to function better, but perhaps not well enough. And so then you can step on the gas of gating and then get those channels to work even better. And that's, again, the vision of what two-drug therapy could look like, where a corrector is combined with a potentiator. So this is some of the science that drove the fact that this could, in fact, work in people. You can see that the CFTR function is shown on the y-axis, and the more drug of this corrector you give as we move from left to right, you can see those orange balls move further up the y-axis as CFTR function is improved by the more corrector that you give, or in this case, the more VX809 that you give. And then in this data, again generated by Vertex, when you add ivacaftor to that, you essentially step on the gas, double its activity, and the more activity you had to begin with, then the more activity that you have at the end. And at the end of the day, you can see on this scale, that was about 30% of CFTR function, which we talked about when we think about G551D patients, might be enough to improve lung function. Because again, we're in that zone where we expect that could result in clinical improvement. Well, there have been results of early testing, certainly not like the phase 3 testing we've talked about with other treatments, but in phase 2 testing, which is a relatively small number of patients, we've seen evidence that this seems to be working. In this study, first the corrector VX809 was given, and that's what you see in yellow in the monotherapy part of this plot. And then on top of that, ivacaftor or VX770 was added to those patients. And that's what you're going to see in the combination therapy zone of the plot that's shown in green. And what we're plotting is lung function over the course of this 2-month study. That's why it's 56 days long. What happened when you got placebo is that really not much. There was certainly no improvement in lung function. If anything, it was drifting down just slightly during the study. And that continued when the patients continued to get a placebo pill or a sugar pill instead of the drug. In the patients that get the highest dose of the corrector, VX809, you can see that there was really no improvement either when that drug was given alone. But then when the ivacaftor was added to potentiate the channels, to step on the gas, that's where this improvement in lung function was observed. So at the end of the day, there's about a 6% difference from those individuals that received placebo. And the hope is that should be enough, especially when the two drugs are given together for the duration and for a long period of time, that could make meaningful improvements in patients with 2 copies or homozygous for the F508 mutation. And that Vertex just announced in the last week that strategy is now going to be tested in phase 3 testing in really large studies that involve maybe 1,000 CF patients across the world to try and test this for a long period of time. These are other results that show that the patients that receive drug, shown in blue, compared to the patients that receive sugar pill, shown in yellow, with one drug really don't have much of a benefit. But when the combination of the two is given, which you can see as the bar graphs in blue shift over to the left, and more patients, each bar graph represents a single patient, has improvement in their lung function, with some having really big improvements. You can see some are up there in the 20%, many are lower than that. But at the end of the day, you can see that if you look at, say, a 10% improvement in FEV1 or a 5% improvement in FEV1, there's getting to be a substantive number of individuals that are having the benefit. So that's, again, another piece of evidence from the studies that suggest that the strategy is certainly worth further pursuit and testing in longer-term trials. So again, based on that, Vertex will do a phase 3 trial that looks at the combination of 809 and 770, or ivacaftor, in patients who have 2 copies of the F508 mutation. And the results could, again, make another big jump in the number of patients that have an effective CFTR drug. And further studies are planned to test the effect of these type of combinations in patients with only 1 copy of the F508 mutation. But further results are being forthcoming, and those are simply in the planning phases. And there are other corrector and potentiator combinations being tested even now. One of them is called VX661, but there are a number of others that are coming online. So I hope I've given you a sense for where the field presently stands, and even announcements that have made in the last week to try and advance CFTR drugs for a much greater number of patients with various CFTR mutations. But what I want to do now is, just in the last few minutes of the talk, is give you a flavor for some of the science that's going on that is attempting to solve the F508 problem by many groups, including both commercial groups as well as academic laboratories, to solve the F508 problem. So what we've shown is that although there's plenty of reason to be hopeful, in the laboratory, even the best drugs only improve trafficking somewhat. And the correction part of that effect is only minimally active with single agents, modestly active with single agents. And the next challenge is to develop a corrector and potentiator combinations that achieve essentially normal levels of function that we would hope would help people for even earlier in the disease process or to an even greater extent. And new discoveries are now pinpointing the problems in processing and the problems in the F508 protein that are enable those type of discoveries to commence. Here's evidence showing that when you use one agent, shown in either the blue or the red, you only get some CFTR function. But when you can combine those drugs together, you can see an effect that's called synergy, or where those effects are more than additive. And you get these large improvements in CFTR function when you use 2 drugs together. None of these are the potentiators. All of these are agents that are trying to correct the folding of CFTR. And that points to the fact that there's maybe more than one step that causes a CFTR protein to fold up correctly. And therefore, drugs are going to be needed to address each one of those steps, or perhaps more than one of those steps, to really make a difference. Well, we know, I think I've given you some introduction to the CFTR structure, and we can break out the CFTR structure into its major parts. In this case, again, the top part, shown in the blue and the red, are the part that makes the channel. And in green and orange, you can see the parts that form the gate or do the machinery of the gating. And it turns out that the F508 mutation is shown with that arrow that says phenylalanine or PHE 508. And it's right in an important junction between where the gating machine works and the pore that energy is conferred to open and close that pore. Well, the other problem that I've shown you is that the F508 protein generates this unfolded protein, especially in one of those green and orange areas, in this case in NBD1, what's shown as NBD1, or nucleotide binding domain 1, a big part of the engine of gating. And when that NBD1 is folded correctly, it looks like on the left side of the screen, a nice tight structure. But when the CFTR protein is not folded, it's a very loose molten structure. And it melts easier. And what we know is that if we can get that structure to form that tighter, more folded structure, we could change its melting point in the lab. So there are groups that are working with pure CFTR to try and change its melting point to address that folding problem of the nucleotide binding domain 1. And in addition, there are people that are using strategies to improve the way the gate engine communicates with the pore, to improve the interaction between those two, because as you see, phenylalanine 508 sits right in the junction between those two places. And so there are drugs that are intended to target the interactions between those two important parts of the CFTR molecule. And this data shows that if you improve the folding, shown on the far left, you get the red line, which is a modest improvement in the amount of CFTR that reaches the cell surface, but clearly not what we'd like to see in normal, which you see in the green line. If you improve the interaction between those domains, you're in the middle curve, and you get what's shown in the blue line, which is a very small effect. But if you do both problems, you get what's shown on the green line. And so that really explains why this synergy has been observed when you use more than one strategy. And it also gives us a much clearer understanding about what needs to be done in the lab to improve the processing of CFTR to get the green line and hopefully land us on the far right end of that green line where you see WT, or the wild type channel, or the norm. That's also another name for the normal channel. So here's I think one of my last videos that kind of wraps this up. Here we can see CFTR again inserting into the membrane. If we can get the situation to be normal, we get this tightly folded structure. Here you can see the purple and the blue nucleotide binding domains becoming their tight structure because the process is normal. The CFTR channel then finishes off its processing, and then all that material comes together in a tightly bound structure because those interactions are present and operating normally and forming the channel that you and I have that are normal functions normally. Because of the science, because of proof of concept in the clinic, and because of these new advances in science, we see now that there are many partners in the pharmaceutical industry that are pursuing drugs that affix these problems. And this is just a list of some, and you can see the various different strategies listed. I'm not going to read them all, just to remind you that there are many different strategies to address each of these problems in the laboratory, and many are being advanced. So I'm going to have a summary slide of CFTR modulators and one vision for the future, and how we've gone from discovery to proof of concept to clinical approval and wide use in a very select population. But studies are already underway that could bring the first drug, ivacaftor, to treat a variety of individuals with responsive CFTR mutations. And the next efforts will evaluate CFTR modulators in individuals with the most common CFTR mutations, including trials that have just been announced. And success has brought additional interest in the approach, including corrector discovery programs by N30, Pfizer, and Genzyme, among others, and you saw a number of others listed. So with that, I hope I've given you a picture of how we can get more and more patients on effective CFTR drugs, and then with the advancement of additional scientific advances that improve the efficacy of these agents even further, how we could then ultimately accomplish our goal to having CFTR drugs for all patients in the CF community, and then finally how smarter, more focused, and more targeted science is enabling us to accomplish this vision. So I know this talk and this journey hasn't been as straightforward as the road shown here, but hopefully I also haven't left you with a street sign that looks like this. And while we know the road is certainly not going to be smooth, and there are going to be bumps in that road marked by successes and failures both in the clinic and the lab, we're hopeful that the road won't look like this. And so with that, I'm going to thank you for your attention.
Thanks, Steve. That was a tremendous talk. We have 5 minutes or so for questions. Dr. Rowe has a plane to catch. As I said, he was kind enough to make a little dent on his busy agenda to come and be with us. So, and we have questions from the web also.
So I have the questions from the web. So Laura, a 48-year-old with CF, is wondering on one of the graphs it showed that normal folks with 100% CFTR function and carriers have 80% function. Does that mean that carriers are symptomatic if they have 80% CFTR?
It's a good question. We're quite sure that carriers don't really have symptoms of CF or manifestations of the disease. And that question points out that once you get over 50, 60, 70% function, you're pretty much normal. And so we don't have to be perfect, but the further we get to that number, the closer we get to 100%, the better the drug should work in the long term.
Okay, and sorry, if you guys are going to ask questions, you need to go to mics, and that'll help everybody that's on WebEx as well hear your questions. So another question, why does he, Dr. Rowe, call severe CF pancreatic insufficient? I have pancreatic insufficiency, but I have mild CF.
So those groups that we've categorized is a very imprecise. And it's only by studying hundreds of patients that you can start to categorize those groups. We know that there's patients who have very mild CF, and we don't fully understand the reasons for that, but CFTR doesn't work in many of their organs, including their pancreas, like this individual that asked the question. But if you look at groups on average, we know on average, again, while there's certainly exceptions, that patients who have pancreatic sufficient CF in general live a bit longer and have a less severe course than the average patient with pancreatic insufficiency. But it's only one component of the great degree of variability among individuals.
Hi, I'm a parent of a child with CF, and I'm wondering what CFTR is.
CFTR is the protein that's caused by the CF gene mutation. And it's the channel that we've talked about that functions to move chloride and bicarbonate.
Hello. You mentioned briefly Adalurin and PTC. Can you give us some information about what the current activity is with the studies on Adalurin?
Right now, it's an open label phase after finishing a very large phase 3 trial. A trend towards improvement in lung function was seen in some individuals, and that was clear in a group of people that were not taking aminoglycosides, which we now know in the laboratory interact with PTC124. And so right now there's ongoing dialogue with the regulatory authorities such as the EMEA, and that's the European counterpart to the FDA, with the company to try and figure out what needs to be done next to try and advance that therapy. And we're waiting on news in that regard.
I would like to know what methods are used to measure improvement. And also, when we get to phase 3, how far are we from the drugs being available to patients?
Improvement in people?
Yeah, the ones that are already being trialed, you say there is 20% of 50%.
There's a number of...
so what methods are being used?
Yeah, very good. So there's a number of ways you can try and measure how a patient's doing. The one that's used most commonly in the largest extensive trials is lung function, but spirometry, who, if you're 6 or older, generally you do almost every time you see the physician. And we often use as a blood pressure, if you will, for patients with CF. That's going to be one marker. The other markers for the CFTR drugs that have been used is sweat chloride. Some of the smaller studies use a test called nasal potential difference. You can actually get CFTR function from the intestines. So some people have their intestinal function measured. And there are other kind of intermediate markers that try and connect what's happening with the protein, CFTR, to what's happening to lung function. And those are things such as what Stanford's been key in advancing, called a measurement called lung clearance index. And others have been using a measure called mucociliary clearance, which measures how well particles move out of your chest, like we've kind of talked about today, but haven't really shown results from. That's often used. All the stuff that I've talked about is generally for smaller studies when people are trying to really understand how a molecule works. And then when you're ready to enroll hundreds or thousands of patients, you try and simplify the study so it can go as fast and as easily as possible.
For people with severe CF, or say, lung function lower than 40 or 50%, what kind of evidence is there that there can be either reversal or stabilization of the damage once they've, started ivacaftor or some of these other treatments?
Yeah. Of course, the ideal situation is that drugs are given before any permanent lung damage could ensue, and I think that's, say, a 10 or 20-year goal for these type of drugs. But right now, what we know is that many of these drugs have been tested, including ivacaftor, in patients who have at least moderately severe disease. And for instance, in the phase 3 study, even patients that are at the lower end of lung function, around 40 or 50%, had as good of improvement as those individuals with higher ends of lung function. So we think that there's good reason to believe that even when moderate problems are present with the lung, that these drugs should be able to help you. In the most very severe form, we really don't have enough knowledge to know do they work quite as well, equally as well, or perhaps not as well because some of the injury is permanent. It's a big reason that we use in our clinic to remind people to do their very best with the therapies that are available, because as these new treatments come online, we think that they'll do a great job of keeping a lung that's not as injured healthy over the long term.
Hi. In that phase 3 study that we're entering now with the VX-809 plus ivacaftor, do we know how long that study is planned for and when we might hear results?
The trial design hasn't been announced, but generally trials that seek approval are 6 months to a year long, and if that turns out to be the case, and then what we can imagine is it will take a couple years for that kind of study to get done, and then the results to be analyzed and then discussed with the FDA so it becomes available.
Thank you, Steve. All right, so while we switch laptops and get things back online, the presentations will be available on the web, not just the webcast, but also the slides. If you like some of those pictures and want to go and think about some of the graphs and discuss them with us in clinic, they're going to be available in our CF Center website. So please keep an eye for those presentations, and we'll be more than happy to go in more detail over any questions that you may have as a follow-up. I know it's a lot of information to digest, but one thing that I've been truly amazed is how over the last just 5 years, the pace at which things are changing in CF is very rapid. So there are many things that we are seeing at CF meetings and then reading in papers and taking our time to digest them and think about them carefully, because again, the field is changing very rapidly. And many of the terms and concepts that we have in the past are also changing as we are getting a much clearer understanding about the disease. So, yes. Yes. Yes, as I said, the slides of all the talks today are going to be posted on the Stanford CF Center website. Give us a couple days to post them, but they'll be there. And again, if you have, as a follow-up from reviewing all this info, it's a lot to digest, questions, write them down, bring them to clinic, email us. We'll be happy to go over all this. Where is my... Oh, here it is. Okay. Here we are. All right, and well, this thing... There we go. So before I start, I also like to, before every talk, we'll do a thank yous to all the ones who help us put this together. It takes more than a village, truly, to be able to put this type of a conference together. I'd like to recognize the support that we receive from the CF Foundation, and particularly from their affiliate, from the pharmacy. They've been kind enough to provide us with funding as an open grant. It's a small grain of sand that helped us to put it all together. So thank you to CFF in particular for the support to a conference. Now, but I'll say back by popular demand, I'm going to present where we are nowadays with our CF Center outcomes. We did this review 3 years ago, and I want to kind of show you where we are in time. And there are a number of things that have changed in the center, in many different aspects, not just in terms of our outcomes, but also in terms of our operation. So I thought it was a good idea, again, not necessarily just by popular demand, but also to bring you up to speed with where we are. Because if you follow from Dr. Rowe presented, and again, it's a lot of information, the future looks more than bright when it comes to CF, and it's not that far out. So we are very much taking a very careful look as to where we are now to prepare for what is coming, which is coming very fast. So as you probably well know, our center has a number of very interesting features compared to the network of centers, not just in California, but across the US. There are about 115 CF centers in the US, and 15 by my last count in California. But there are a few things that I'm highlighting on this slide that really distinguishes us. Probably the one for which we're very well known is that we have the largest center in Northern California. We have over 400 active patients, and the number keeps growing by the year. So we're very much on track to be one of the largest centers in the country in 5 years from today, if the pace continues. We also have the largest CF program in California, and not just in terms of volume of patients, but also in terms of the size and the activity of the team. We have a very solid team working with the adult patients. And we have strong support from the institution at all levels, being it Packard or Stanford Hospital or Stanford University at large, are very much behind all the efforts in making the program grow. And less than 2 years ago, without wanting to, we became the only CF center with an active pediatric lung transplant program west of the Rockies. We're the only place in all that multi-state region that can do pediatric lung transplants. Now, we were the only CF center in California to have a translational therapeutic development network designation. That was thanks to the effort of many of the people that you see on red shirts walking around. So give them a pat in the back for that. And because of all these features, we become pretty much a safety net for the region. We see patients with all levels of disease severity and all sorts of unanswered questions that come from all of California and the Sierras and Southern Oregon. So we cover a pretty wide geographic area, which we're very happy about because, again, we're feeling that it's part of our mission to provide that safety net and help people that are not finding the right answers for their problems. So I'm giving you this as a background so you can understand a little better our outcomes. You need that sort of contextual understanding as to what is it that we do, what is it that we try to do for our patients in interpreting the data. Now, I always like to show this, that as a CF center, we're not just a clinic doing CF care. We are a very sort of complicated organization. Our mission includes caring for patients, being very active on the research that hopefully will provide a cure sometime soon, and also teaching and training the future leaders in CF. And all these activities are very much intertwined. You know, it's a very tightly neat organization. And we have clinical programs that we try to keep as close as possible with patient advocacy groups and patient representatives, because we're very much interested and wanting to hear your feedback and pay attention to your concerns. So this is how we look over the last 7 years or so in terms of how things have been changing on the center. And again, our patient volume has been growing at a very steady pace, slowed down a little over the last year or so, and this is up to the summer of 2012. So it's not as up-to-date as I would have liked, but just to give you a flavor for, you know, how big we're getting and at what pace. So when it comes to looking at the outcomes, we like to do it by specific groups. And not necessarily because there's an adult center and a pediatric center, but because we know age is an important factor when it comes to CF, and particularly when it comes to outcomes. And most of the data I'm going to show you is what we get from the CF Foundation. They are the ones who are processing this information and feeding it back to us, meaning it's not us, you know, trying to gather the data in some disorganized way or trying to massage it. You know, this is what we get from the CFF as a report of how we're doing. So that's what I'm sharing with you, trying to be totally transparent as to how we are being seen by the rest of the network. And all this information is available on the CFF website, by the way. If you wanted to get more detail about some of the numbers, you can go to the CFF website, look under Care Network, and you'll be able to see the outcomes. And not just for Stanford. You know, if you're techie enough, you can make a graph, a bar graph that will put the outcomes of all of California or all the West Coast or the whole US. So you can really compare, you know, where we are, how we're doing compared to the rest of the country. So I'm going to give you just a quick digest, and because I really want to save time at the end to have some dialogue about this. So we know nutrition is very important in CF. It's the first thing that pops up on our outcomes report, and it's the thing that the CFF pays very close attention to, because we know nutrition is very important. And this is where we stand nowadays. And it's divided by the newborns, because the newborns can primarily through newborn screening nowadays, meaning they're diagnosed at birth before they're symptomatic. So our median weight for length, meaning how plump a baby looks, is placing us somewhere towards the lower end when it comes to the rest of the country. And when we look at how we've been doing over the last 5 years, because newborn screening has been implemented in California for the last 5 years, since in 2007. The summer of 2007 is when it was implemented. And if you're looking at the first 2 years of life, then you need a few years from the time that was implemented to start getting a feel for how we're doing with these babies. And what is apparent is that we are having some challenges in dealing with this very early patient population, in the sense that their nutritional outcomes are not truly where we like them to be. The blue line is the ideal, the green line is where the average for the country stands, and the red line is where we are. Now, when it comes to the older age groups, from age 2 to age 19, meaning about or around the time when they have moved to the adult program, this is where we are. And with this, we're very happy because we are pretty much on the top tertile for the country. And if you look at it, it's not a product of an overnight effort. This, and unfortunately the graph is chopped in 2007, but if I show you where it started in 2005, there's been a slow, steady improvement in our nutritional outcomes, so that now we are above the national goals when it comes to, and here we're showing median BMI as an example of how to look at nutritional outcomes. So again, it's been and I have to thank and recognize Julie, who's sitting somewhere back there, for her resilience, because it's truly many years of keeping at it that is moving us where we are nowadays. And if you use your eyes to connect the dots, you could tell, well, maybe next year we're going to be much closer to a blue line, which has been on the top 10. So this is, again, a recognition of Julie's resilience. The red line means that 100% of the patients are having time with her to do the nutritional counseling and evaluation. So on that metric, we're well above what the national average is, which is about 90% of the kids are being seen by a nutritionist. So it's paying off well. And the interesting thing is that, they're obviously one of the first reactions we had 10 years ago when we started thinking with the CFF as to how to make improvement on some of these outcomes is that maybe we need to be more aggressive with nutritional supplementation and things like tube feedings. And if you notice, at the same time that our outcomes have been improving steadily, we haven't really jumped up on the use of nutritional supplements and gastrostomies and things like that. If anything, compared to the previous year, it looks like we're a little below where we used to be. So that means that, we've been able to figure out how to get you guys to work on this without having to be much more aggressive than what we already have on hand. But it also makes you wonder as to, if we have more room for improvement there if we were to step a little harder on the supplementation and the aggressiveness of the nutritional support. Now, when we look at the adult patients, and here the CFF reports it at age 20 and above, and that's mostly because of the BMI percentiles. They apply only until age 20, and from then on it's just the actual BMI, which is a measure of body mass, meaning how plump you are. And the ideal is to, or the goal is to have patients above 22 as a BMI number. So we sit a little below the national goal, and certainly far from those places that tend to have more, what we can call it, plump people. But the good news is that, again, like with the pediatric patients, we've been slowly moving in the right direction. We used to be below the national goal, and now we're right on the national goal when it comes to the median, meaning how our average CF patient is doing on the adult center. We're very much right on the national goal, which is the green line. And again, thanks to Laura, we are 100% when it comes, do your patients have access to a nutritionist that can help them, blah, blah, blah. Well, yes, we're 100% on that metric. And if you notice, there's a big drop-off. There are many places where it's really hard to get a nutritionist to be able to assess a CF patient, believe it or not. And, we have stepped up on the use of nutritional supplements for the adult patients. And again, makes you wonder if you press a little harder, where we're going to get those outcomes, So there's a lot of room for improvement. Now, the big one in CF is the lungs. Pulmonary function is one of the many metrics that is followed very close for this. And the FEV1, meaning the forced expiratory volume in the first second, is the number, because we know it's tied to many different things, not just how the patient feels, but also morbidity, mortality, exacerbations, hospitalizations. It's very much tied to FEV1, so it's a very good predictor and a very good metric. Unfortunately, we cannot apply it before age 6, so the data we get is from age 6 to 12, meaning from 6 to adulthood, and it's broken down by age groups. So if we look at the preteen group from 6 to 12 years, we're standing below the national average. And our numbers have sort of just crept up and down below the national average on that specific age group. Now, if we look at the teenagers, believe it or not, we're in the top 10 when it comes to pulmonary function among teenagers. So we need to think a little more about what we're doing with the teenagers that is working so well and see if we can translate it down to the younger age group. And if anything, if you notice, is over the last, and again, the graph here, if I pull it back to 2005, is over the last 7 years that we've been slowly but surely making a good dent on the pulmonary health of our teenage patients. So right now we're in the top 10 nationally for that value. When we look at our adult patients, and the adult report now is broken down on younger than age 30 and older than age 30, because if you have gone past age 30 in CF, there's probably a lot of meaning on that. So that's why it's now broken down on those two categories. So we are very close to the median when it comes to the pulmonary function between age 18 and age 30. And if you look at the patients older than 30, when you'll think, well, things at age 30 is probably when they start going south. If anything, the pulmonary function metrics for those patients are in the top 10 nationally. So again, maybe from the older groups, we can learn things that will help us with the younger groups. Now, the other metric that is quite impressive is when you look at the exacerbations or hospitalizations. We are one of the places that has a very low rate of exacerbations in the pediatric patients. Our rate right now is about 20%, which is very much below what the national average is. And with adult patients, we're also just below the national average when it comes to rate of exacerbations. Now, if you think about these two things, you'll say, well, maybe we're getting things to look better because we're using a lot of antibiotics, or we're dealing with, we're pushing for more severe infections, but our numbers when it comes to Pseudomonas infection, particularly in the pediatric patients, actually look very good. We have a fairly low rate on, and looking, and this is, again, you need to think that this is all news because it's from 2 years ago. The report takes over a year to be generated. But just taking a quick look at how things may look like for the next report, we are not just keeping that number, but it looks it's even dropping down some more. So we have very good success in not just treating the pulmonary disease, but also keeping people away from the chronic Pseudomonas situation. Now, these are the outcomes when it comes to health outcomes, but the CFF is also paying very close attention, and we are all paying very close attention. So we call process outcomes, meaning are people getting the care that we know gives you the best results. So one of the things that this track is how frequently people are coming to clinic, and the current recommendation is that kids with CF should come to clinic at least 4 times a year. And this metric has been watched very closely by the CFF. Because it's part of the care guidelines that kids with CF need to be seen with certain frequency. And what we're seeing right now is that we have reached a really good point. About 75% of our patients are being seen in the clinic at least 4 times a year. And I'm very pleased with that. And obviously, I like to be like this guy here. It's 100%. But I can tell you, if you look at historically where we were, where we're going, we are making very fast move on that. And again, this is more all news. What is coming for 2012 looks even better. And it's not a product of an overnight thing. You know, I mean, it's, if you notice the trend, it truly started in 2009, when we really started making a dent on being able to give you access to what you needed. In terms of the adult patients, the same thing. We used to have really low numbers. Now we're above the national average. But, you know, I also want to be an overachiever and see if we can get a little closer to what the high-achieving centers are doing. And notice that, you know, for adult patients, it's not 100%. Even the people that are trying their hardest and work the closest with their patients cannot get them to clinic as often as we like to see. So a lot to learn from all this. Now, among the many other things that we monitor in CF patients, you know, I did part of my career studying diabetes in CF, so I've always been a big pusher and advocate for glucose screening and diabetes screening. We make a lot of move on this. It's something that we're always falling behind. A lot of it, I think, has to do with logistics of getting the testing done, but we're getting better at that. So now we are very much at a national average, meaning about 80% of the CF patients are getting at least once a year, getting on a yearly basis their glucose screening done. So I think the last time that I did this sort of a presentation, when it comes to plotting glucose screening versus clinic visits, because obviously if you don't come to clinic, you're not going to get your order or your request to get the glucose screening done, so these things are all tied together. And this is one of the ways in which the CFF is looking at these metrics, you know, doing these sort of match comparisons and trying to correlate what is tied to what. So back in 2009, we're not doing that well. If you notice, we were down here, and the blue and all the dots there represent, I was looking just at the region, not at the national, because then I'll have too many dots. So if you look just regionally, we were like the laggards, you know, we were like way out, because the goal is to have 100% for both, which is all the way up here. So when I redid this thing, I'm very pleased to see that, well, you know, we have moved very close to that goal, and we're very much getting ahead of most of our neighbors now. But, you know, it's taken us 3 years, or more than that, really, because the 2009 reflected the previous year, so it's 4 years of just keeping on it, but not just being insistent, you know, but also reviewing our processes and trying to make it easier, not just for you, but also for us. So again, these are some of the highlights, just to summarize. You know, we now are among the top 10 in the US for pulmonary function in teenagers and people older than 30. And those are your most challenging groups. So that means that we have figured out something that works really well. We just need to get smarter at dissecting out what are those pieces that we can bring to the other age groups, so we move them all up. We are certainly now well above national goals for the nutritional parameters. In children and in adults, we just hit the mark when it comes to being within the national goal. We're doing really well with diabetes and glucose screening. And I'm very pleasant, I won't call it surprise, I anticipated that because of how aggressive we are with care. We don't have high rates of infection, and I didn't show the rest of infection graphs. But there are all these germs that have funny names that scare us in CF, and we don't have high rates, we have very low rates of those germs. So we're doing very well with our infection control. So I always like to quote Sir Robert Giffen, who came up with this statement in a debate back in the 19th century in England about allocation of resources, and people were talking about statistics and trying to make predictions, and he said that there are 3 degrees of comparisons in lying. There are lies, outrageous lies, and statistics. And here's where the lies, damn lines, and statistics phrase came from. And interestingly, in Parliament, when people were debating, I think it had to do with budget, like nowadays, and people were quoting him, Lord Leonard Henry, who was pushing some law, responded to him by saying, okay, well, after all, facts are facts, and although we may quote one to another with the chuckle of the lies, damn lines, statistics, still there are some easy figures the simplest must understand and the astutest cannot wriggle out of, okay? So, well, understanding the data, right? First, you can say, well, the things that don't look good, they're wrong. Maybe we didn't enter in the database things, or the CFF is not getting really what we are really doing, or there are mistakes, right? The next thing is to say, well, yeah, it's right, but it's not our fault. It's the hospital, it's insurance, it's something else, right? And the next is to say, well, okay, it's what it is, it's our fault, but we cannot do anything about it. It's what it is. CF is a disease anyway. And I think what we are as a group, and we're very much, and again, please give a big pat in the back to all the red-shirt people around you, is that, we've taken this very seriously, and we are very much recognizing that it's only our work with you what is going to help to correct these, I won't call it problems, more like push it to the next level. And we start by recognizing that, every system is perfectly designed to achieve the results it gets, right? Karma, meaning what we get is because of what we do. So we need to take a systems approach to it. Because, these guys, they're far gone, meaning if you keep doing things the same good old way, you're going to be left more than behind. So there are a number of things that we're doing. If you have noticed, there's a lot of change going around the Packard and Stanford area. There's dust and construction all around, and I think that's pretty symbolic of what is coming. It's not that we're just building new facilities or beautiful ivory towers. There's a whole redesign that is going behind the building effort, meaning we're not just going to have space. We want to have new processes in place. And more than anything, to catch up with where the 21st century is, particularly where medicine is going. We're talking about genetic-based prescriptions based on your genotype. We're going to prescribe, we're going to monitor new treatments coming up, new ways of monitoring whether those treatments work or not. We're going to have to think about what we're doing nowadays, how we're going to adapt to all this, and not just adapt, but get ahead. So one of the things that you may have heard is that we're going to move to a new clinic space that is being remodeled for us. The timelines get pushed a little. It'll probably be sometime during the summer. And again, we're going to take it as an opportunity for not just having new space, but also having new processes in place, redesigning how we do things. And my colleague Sumit Bhargava, who I don't know if he's around here, he's taking a major lead on this, for the whole pulmonary group, trying to redesign clinic flow, and take that again as an opportunity to rethink how we do things. With Mary, we're engaged, along with Kathy and Paul Mohavir, and I don't know if Paul is here yet, but we're engaged on this initiative that has been sponsored by the CF Foundation to create leadership course, meaning I'm not going to sort of direct the center as a dictator. We're going to try to do it more by committee and engaging all of our colleagues on this process. We're trying to think carefully as to where to go next when it comes to education. Our first talk today left us all with a head that is full of stuff, and how we're going to process that, how we're going to individualize it to the specific situation of every single patient. Go back, from thinking and treating the average patient to thinking and treating the patient that is right in front of me. We need to go back to that. I was asked to write a review paper on where CF is nowadays and all that, and as I was doing my review of the literature, I came across something that I found really interesting, which is that next month is going to mark the 10th anniversary of the completion of the Human Genome Project. And if you read the stuff that was put out 10 years ago, when this was completed, people were talking about the things that we're nowadays living in CF. But very little of it has translated or been applied or happened to other diseases or just medicine in general. But if you read what people were saying back then, they were pretty much laying out a vision for how medicine will be 10 years from today based on this very powerful knowledge that had just been completed. And the way I see it, it's only in CF that we've been able to realize that fully. Not only now we have therapies that are addressing the basic defect, who, if anything, have demonstrated to us that it can be corrected, that CF can be cured. But on top of that, all these buzzwords about genomic medicine, translational medicine, personalized medicine, individualized care, we're very much right there when it comes to CF. We just need to get better at realizing where we are and moving forward from there. So this summer I'm going to, one of my, as director, obviously I need to do my yearly strategic planning and things like that. So I'm going to propose to my colleagues to engage in a process of laying out what I want to call the transformative CF Center strategic planning, which is how to get ahead of the wave again, because we've been very much ahead of the wave, and I'm not a surfer, but I like the concept of being, ahead of the wave. And all of that obviously has to include multiple partnerships, with you guys, number one, because you are our primary consumer, but also with organizations like the CFF and CFRI. With CFRI, we've had some good conversations as to, for example, how to do innovative education, and we've talked about where the gaps are, how can we best meet the needs of our Spanish-speaking populations, for example. Nowadays, a third of our patients, and we predict probably over that, probably close to 40% in the next 5 years are going to be only Spanish-speaking. We need to catch up with that and provide better educational materials to our patients. As well as, with our community at large, scientific and patients and advocacy groups and insurers, because it's sort of what is coming. We're going to be asked to look more carefully at what we're doing, and not necessarily for cost containment, but for cost efficiency. So I'd like to stop there and leave maybe 1 minute before we take a break for questions, but as I said, this is a two-way street, and I'd like to spend the day talking to all of you guys, hearing your thoughts, and this is an ongoing process. Thank you.
I do have one question on the statistics. Apparently, we have one of the most diverse centers. Mm-hmm. Does that have any impact on the statistics? Do they have that into consideration? Because I hear saying that some people have a stronger degree of CF than others.
Yeah, those things are somewhat adjusted. What I show you is the actual raw scores data. What you see on the CFF website has been adjusted. That's what they call. There's some adjustment that is done, but it doesn't really take care fully of the things that you're bringing up. And I don't know if you had the pleasure of meeting Dr. Mimi Boo. She's somewhere in the crowd. She's one of our junior faculty. She's a CF care provider, and she's very interested on the diversity issues, Is CF on a Spanish-speaking person from Mexico the same thing as CF on someone that has Danish inheritance, There's a lot of data to suggest that it's not the same thing. And again, we want to sort of go back from talking about the average person and the group of outcomes to just the individual, because it makes a huge difference. Okay, thank you. So we're going to take a break. And again, I'm around all day, so please grab me. I'd very much like to hear your thoughts about all this. We'll reconvene at 10:45 for our next session. Oh, sorry. I skipped something. Yeah, we'll do that. So we'll reconvene at 10:45 to continue with the session. We'll try to condense the next 3 talks. Yeah, no, but I think they need a restroom break, please.
Is yours?
Yeah.
I was wondering if I talked to you when I was driving. Did I give you a call? I don't remember anything. I forgot. I think I did call. And you didn't want to, I don't know, maybe. And you had another boyfriend. Do you remember talking to me or not?
I talked to a few people, like over the
So
years.
it's like so
People have
many.
given,
Yeah.
out my
Your phone
number.
number. Yeah.
But it sounds like I think that
I was wondering the other day, I forgot to share a because that week was so busy.